The Stephen Paget Memorial Lecture celebrates the life of Stephen Paget, who passionately believed that a greater understanding of physiology would lead to better medical advances. He was the founder of the Research Defense Society, which later became Understanding Animal Research. This year, we're delighted to celebrate the 88th lecture, adding Professor Dame Sarah Gilbert to our long and eminent list of lecturers. Professor Gilbert is principal investigator at the Pandemic Sciences Institute, an interdisciplinary research institute at the University of Oxford, with the mission to discover, create, and enable practical, science-driven solutions to infectious disease threats worldwide. She initiated and led the developments of the Oxford AstraZeneca Covid-19 vaccine in 2020, one of the first vaccines against the SARS-CoV-2 virus approved in the UK. It's been used in over 180 countries and is estimated to have saved more than 6 million lives worldwide. Professor Gilbert's primary research interest is the development and production of viral vector vaccines, from initial concept through clinical trials onwards to rollout. She currently leads vaccine development for influenza as well as for several emerging pathogens, including influenza, Nipah virus, Middle East respiratory syndrome, and Lassa virus. Professor Gilbert was made Dame Commander of the British Empire in 2021 in recognition of her work during the pandemic. Her book, Vaxxers, describing the development of Vaxavar, I will mispronounce it, I'm sorry about that, was a Sunday Times bestseller. And I'd like to finish my introduction by sharing a quote from an interview Professor Gilbert gave UAR recently. Animal research is essential, but we are always looking for opportunities to reduce the number of animals used, whether that is through very careful experimental design or statistical optimization. There's an increased focus on using human organoids to test vaccines and look at immune responses, but these are not fully ready to replace animals in the research we conduct. Animals are still very much needed. Tonight, I'm delighted to introduce Professor Dame Sarah Gilbert and her lecture, Development of Vaccines Against Outbreak Pathogens.
Thank you, and thank you for coming this evening on this very dull, dreary, wet evening. I'm sure you'd would all rather be somewhere warm at home, but let's get on with the lecture. So I want to take you back to something that happened in 2014, which I still remember very clearly, but may not be so familiar to all of you who don't work in this field of outbreak pathogens. So the Ebola outbreak in 2014, which happened in 3 countries in West Africa, Liberia, Sierra Leone, and Guinea. This was the largest Ebola virus outbreak that the world had known. It certainly wasn't the first one. It was the virus first discovered in Central Africa in 1976, and it causes a hemorrhagic disease, and it can be dealt with if it's dealt with quickly by contact tracing and quarantine. So you find somebody who's ill with Ebola virus, you find everybody they've had close physical contact with, you quarantine all of those, and you wait until you know that they have not developed the disease, or if they do develop the disease, then they receive treatment. There's no specific treatment, it's just supportive treatment until they either die or recover. And that works well when the outbreak is in a rural part of a country where there are few personal contacts between people, and it's quite easy to trace those who have been in contact with the person who goes on to develop Ebola virus. What happened in 2014 was different. The outbreak actually started in Guinea at the very end of 2013. It was thought a boy was playing with a dead bat under a tree and he became infected, because this virus is carried by bats, and bats have rather strange immune systems. They're quite different from ours. Rather than trying to overpower an infection and rid it from the body, they just coexist with it. So their immune system is very laid back, very relaxed, and they often carry a lot of viruses, and they seem to do no harm to the bats themselves, but they can be passed on to other species. And that's what happens with Ebola virus and other filoviruses. Marburg, Sudan virus are very closely related viruses. And an outbreak started in December of 2013 and it spread, and this time it wasn't contained. It rumbled on and it spread and it ended up crossing borders and importantly getting into cities where there was a much higher population density, more interpersonal contact, much harder to trace those contacts. And the numbers slowly grew through the spring of 2014. And then when it got to the summer, they really started to increase a lot. And at this point, in July, August of 2014, the world started to take notice because this was becoming a threat to the rest of the world. We were starting to see cases spread outside these three countries to neighbouring countries in Africa, such as Mali, but also a few cases of people travelling, turning up in the US, in Texas, being infected with Ebola virus. It wasn't recognised very quickly. The nurse became infected. Also, some healthcare workers from Europe were repatriated because they'd been infected, and sometimes there was onward spread of infection, although it was contained relatively quickly. So this was starting to become a problem. Armies from various places were going in and building healthcare facilities, places where supportive therapy could be given to improve the chances of survival, and also to quarantine people and try to stop the ongoing contact. And in Oxford, we were asked if we would help develop a vaccine against Ebola virus, which work had started on. It was part of a US biodefense program. It had been tested in non-human primates and shown to be protective against Ebola virus infection in those non-human primates. It had not yet entered clinical development, so no human had been vaccinated. A batch of vaccine had been manufactured, ready to start clinical trials, which was happening on a fairly slow timescale. Hadn't been put into the final vials yet, so that's the last stage in the manufacturing, and those trials hadn't been planned. It was an adenoviral vectored vaccine based on a simian adenovirus called ChAd3. And the only place in the world at that time that had conducted a clinical trial with a ChAd3 vector vaccine was Oxford, because in our clinical trial unit, we had partnered with the owners of the ChAd3 technology to test, it was actually a hepatitis C vaccine, using the ChAd3 technology. So we had had the experience of applying to our ethics committee, our regulators, the MHRA, for permission to conduct this trial. And we'd had the experience of conducting the trial. We saw what dose we were using. We saw what reactions people made to the vaccine, whether it was local reactions like soreness at the injection site or systemic reactions like a headache or a short-lived fever. We'd been through all of that. And so we were asked, because you've done this before, and your regulators have seen this before, and your ethics committee have seen this, could you please take this vaccine, arrange to have the final filling into the vaccine vials done, and then conduct a trial as quickly as possible, please, so that we can then move this vaccine into the outbreak area and see if it actually protects people against Ebola and start using it to contain this outbreak. So we agreed. We were asked to do this in August, and it took us a month to get all the approvals in place, and we started vaccinating people. We vaccinated 60 people. We did a month's follow-up. We got all of the safety data, all of the immunogenicity data. We showed that the safety was as expected. There was some mild to moderate, short-lived adverse events, the sore arm, maybe a headache, those kind of things. And we saw strong immune responses after a single dose of the vaccine. So we then handed this on to a group of healthcare workers in Mali, next to the outbreak area, and they did another trial. They repeated what we'd done. They showed also in an African population, very similar reactogenicity, very similar immunogenicity. This was ready to go into efficacy testing in the outbreak. But that didn't happen. So by November, we thought we've done everything we can. We've really pulled all the strings. The regulators have been amazing. We've got everything done. This is ready to go into testing in the outbreak area. And at that time, at the end of 2014, there were still large numbers of cases in 3 countries, well, particularly in Liberia and Sierra Leone, but also quite a lot of cases in Guinea. What happened was a protracted debate about the ethics of conducting a vaccine efficacy trial with such a highly fatal disease. Now, this was an important debate to have, but in my view, it was happening far too late. It could have happened the previous summer. And the arguments went something like this. If you test a vaccine in an efficacy trial, you have to have a control group. The gold standard randomized placebo-controlled trial. Some people get the vaccine, some people get a placebo. You follow them, you see who's infected, and you see if your vaccine actually works. And you count the number of cases that accrue, and everybody's blinded. And when you get to a certain number of cases, and this is how we did it in COVID, when you get to a certain number of cases, you then say that's enough to do the statistical analysis. We'll break the code. We'll see which of these cases come from the placebo group, which come from the vaccine group, and then we'll know how effective the vaccine is. That's how you normally do these kind of things, but this is a highly fatal disease. So the argument went, we can't do a placebo-controlled trial with such a highly fatal disease. We can't give people a placebo, recruit them into a trial and give them a placebo. But if we don't do some kind of trial, we won't know if the vaccine works. If we just start vaccinating everybody, we won't know if case numbers start to drop. Is it the vaccine or is it just happening anyway? And we really need to understand if this vaccine works so we know if it's worth using to end this outbreak and also to use in future outbreaks. So there are other trial designs that were discussed. So one is called a step wedge design. And this hinges on the idea that you can't vaccinate everybody across these 3 countries in the outbreak area all on the same day. So what you do is you start your vaccinations at one side of the outbreak area and you gradually move across and vaccinate as you move across the area. And what you're expecting to see is as you move across with your vaccination campaign, your number of cases starts to drop. And that means that you're vaccinating everybody as fast as you can, and you're concentrating your efforts starting at one side and then moving across in a way, because you can't vaccinate everybody on the first day anyway. And that's fine, but statistically, it's really very difficult to understand what's going on if you do that kind of trial. And it's estimated you need to have 10 times more cases in order to understand if your vaccine's really working, because it's quite difficult to interpret data that comes from that kind of step wedge design. So the third design that was discussed was a ring vaccination study. And this involves identifying a ring. So you have a case, you have the contacts of the case and the contacts of the contact, and those people form a ring. And you identify lots of rings. And then you randomize the rings to receive either immediate vaccination or delayed vaccination. And if it's delayed vaccination, they get their vaccine 3 weeks later. So this means some people have the opportunity, if you want to put it that way, to be exposed to the virus during that 3-week period, and they may then develop Ebola virus disease, whereas other people are getting vaccinated immediately, and if the vaccine protects them, then they won't. Now, this is considered more ethical than just not vaccinating people, because most people wait 3 weeks, and they're going to come back and they're going to get their vaccine. They won't be left unvaccinated. Now, with any kind of placebo-controlled trial like this, if you have a really high vaccine efficacy, you don't need a very large number of cases within the trial to know that the vaccine worked. It's something between 15 and 20. It's not very many. And these trials were taking place where there was a lot of healthcare support. The hospitals had been set up, so if anybody did get infected, firstly, they would be diagnosed very, very quickly because they'd be followed. And the sooner you can find out that somebody's infected, the better chance they have of surviving that disease. And then you can get them straight into your hospital and give them the best supportive care that you can and greatly increase their chances of surviving the disease. But it means that some people will still be exposed to the virus. As it turned out, when this trial design was used, and it didn't start until April of 2015, sufficient numbers of people actually refused vaccination that actually, with hindsight, the number of refusals could have constituted the control group. But that's probably also not a very good way to design your vaccine studies. So the delayed ring vaccination strategy went ahead, but by this time, by 2015, April of 2015, the number of cases was really declining. And the opportunity to conduct clinical trials of more than one vaccine was lost. So originally the plan had been to test the vaccine that we'd helped to develop, the ChAd3 vector vaccine, and another vaccine, another viral vector vaccine, which Merck had taken on and prepared for the clinical trial. That's the VSV vector vaccine. We thought that both of them were going to efficacy testing, but actually in the end, only the VSV vector vaccine did go into efficacy testing. And good news, was found to have very high efficacy. It's not the ideal vaccine to use in West Africa because it requires ultra-low temperature storage, -80 storage. The manufacturing process isn't scaled up, it's manufactured still in roller bottles. Whereas the ChAd3 vector vaccine can be stored in a fridge, and there's a very large-scale process, and this when that's part of the technology that it's very similar to the technology Chavix 1 that we developed in Oxford, and we used it in the pandemic. And that has a lot of advantages, and that's why we use it, because it doesn't require frozen storage, and it's possible to manufacture literally billions of doses, and we did that in 2020 with AstraZeneca. So we felt that a vaccine, not our vaccine, a vaccine we'd helped develop, but a vaccine that had very useful characteristics didn't actually get the chance to go into efficacy testing. Johnson & Johnson also had a vaccine which they started testing a little bit later on. That was eventually licensed, not through efficacy testing, but through immunobridging, which is something I'll tell you about a little bit later. And these vaccines only cover Ebola Zaire, so there's also Sudan and Marburg virus, other filoviruses. So we've got a vaccine now, the Merck's BSV Ebola vaccine, got one, but we're going to need more. So I told you that there was this delay to start the efficacy trials because there was debate about the ethical way to do it. So we felt that the efficacy trials were probably going to start in December of 2014. And in fact, they didn't start until April of 2015. And these are the deaths in Sierra Leone, in Liberia, in Guinea. Now, this spike goes up to 1,200 deaths in a month in Sierra Leone. Now, we probably couldn't have done anything about that with the vaccine because we weren't ready to start testing until that month. But here we've got more than 400 deaths in Liberia in February, more in April. Had we started testing the vaccine earlier, we would have been able to present some of these deaths with either of the vaccines, most likely. But instead of waiting till April and then test the vaccine and discover the vaccine works, but actually the outbreak's been contained by other means now. It has been used in subsequent outbreaks, but there was this challenge around the fact that people were still dying and we were waiting to test the vaccines because the world wasn't ready to do this. The infrastructure wasn't there. The plans hadn't been made. And this outbreak was widely recognized as a failure in outbreak response. It really wasn't good enough. And following that, a lot of organizations started to put together their list of priority pathogens, and this is the one from the WHO, but other organizations have very similar lists. So this is simply a list of all the viruses that we know have caused outbreaks in the past, and like Ebola, are likely to come back and do it again. And if we're not ready, if we don't have vaccines and diagnostics and therapeutics, we're going to be in this mess again, and it gets very expensive in terms of human life, but also in terms of money, trying to scramble to contain an outbreak. If we're better prepared, if we have things ready to use as soon as an outbreak is identified, and we have the clinical trial protocols, importantly, that we have agreed that we're ready to use, we could shut down these outbreaks earlier. So this is the list of viruses, and they've been identified even going back to 1947 for Zika virus, and that was in the news much more recently. Rift Valley fever virus is on this list, whereas that was identified in 1931. We have a vaccine for sheep against Rift Valley fever virus, but nothing for humans. So the idea was to start developing vaccines against all of these, have stockpiles, and be ready to respond. Since then, the approach has moved on slightly. We're now, instead of just talking about the priority pathogens, we talk about priority families, because these viruses form into different families. So SARS-CoV-1, SARS-CoV-2, MERS, these are all coronaviruses. Problem we had in 2020, when we had a pandemic caused by a novel coronavirus, is we didn't really know what we had to do to make a successful vaccine against a coronavirus, because we don't have any other vaccines that are effective against human coronaviruses. There are 4 so-called seasonal coronaviruses that regularly infect people. They give a toddler a cold, snotty nose, they're a bit off colour, then they get over it. And we can be infected throughout life, but it's just a cold, and we don't know whether it's a seasonal coronavirus or something else. So we don't pay much attention. And we don't know what we would need to do to vaccinate with a vaccine to prevent those infections. So not knowing how to vaccinate against a human coronavirus made life difficult for us in 2020. We have other types of viruses here. Marburg, as I said, is a filovirus like Ebola. We have bunyaviruses, we have arenaviruses, and the idea now is that rather than concentrating on the individual pathogens, which have been identified at different times, at different areas of the world, we think about the viral families, because if we understand how to make a vaccine against a member of a viral family, that gives us a big head start with having to make a vaccine against any other member of that same family, which may not be one that we've seen before, but if it's a close relative, we know what we need to do. So now, if we have another outbreak with another coronavirus, we've got a much better understanding of what we need to do to make a vaccine. So from 2015 onwards, an organisation called CEPI formed, Coalition for Epidemic Preparedness and Innovation, and they received money from many governments and the Wellcome Trust and the Gates Foundation to start funding the development of vaccines against some of these pathogens. And their first call for proposals was vaccines against MERS, Nipah, and Lassa. And together with Janssen Vaccines, part of Johnson & Johnson, I led a bid to CEPI to make vaccines against all three of the ones that were first on their list. And I'm going to tell you a bit about those now. So the first one is Lassa fever virus, another African virus. This isn't really an outbreak pathogen. It's endemic in West Africa. Lots of cases in Nigeria, now year-round infections in Nigeria. Now, unusually, this is not a virus that commonly transfers well between person to person. That can happen in healthcare settings. It's very dangerous in pregnancy. And if a cesarean section is performed to save the life of a mother, there will be a lot of Lassa-infected blood released, and that is very dangerous to the healthcare workers who can become infected and have died of Lassa infection. But actually, most cases are through indirect contact with this little multi-mammate mouse, the Mastomus natalensis. And these carry the virus. They're very shy little creatures, but they like to get into people's houses, get into their food stores, steal the food, and they contaminate the area with their droppings, and that gives you Lassa virus in dust, and when people clean up, or if they have contact with the rodents, they can be infected with Lassa fever virus. And most infections are rodent to human, not human to human. So dealing with the virus infection in the rodents will be excellent, but nobody does this. Now, it would be an interesting thing to try to do this. These mice, they're quite cute little creatures. Apparently, they're kept as pets in Northern Europe. In Germany, in Scandinavia, it's not uncommon to have these kept as pets. It will be possible to develop colonies and start testing vaccines in the rodents, because if you could clear it from the rodents, then the humans wouldn't be exposed. But this isn't something that's been done yet. It would have to be an oral vaccine, and you'd put it down as bait so that when the rodents came into the food stores, they'd actually get some of the vaccine. That's quite a lot of development needed to do that, so we're not really ready to do it. So instead, we're developing a vaccine to use in the people. So we've done some testing of the vaccine, and the Lassa fever virus is infectious and fatal in guinea pigs and non-human primates. So we made our adenovirus vector vaccine expressing the external glycoprotein of Lassa virus, lineage 4, and we vaccinated some guinea pigs. This was done in the US at Rocky Mountain Labs, NIH, and the guinea pigs had either 1 dose or 2 doses of the vaccine, or they just had a control. So we can do placebo-controlled trials in guinea pigs. And this is the survival. So if they had 1 dose or 2 doses, they survived. If they weren't vaccinated, they didn't. You can see the temperature increase in the control group, whereas those who are vaccinated, either 1 or 2 doses, very steady temperature, and the viral loads present only in the unvaccinated animals, whereas it was down on the baseline in the lung, liver, and spleen of the vaccinated animals. So it's a very good vaccine for guinea pigs, which is a start. And we've also been able to use the guinea pigs to look at heterologous protection. So there are 7 different lineages of Lassa virus. And we're using lineage 4 to vaccinate, but we've been able to vaccinate with our vaccine and then challenge with a virus of a different lineage to see if that protects, and the answer is it does. Unusually, Lassa fever virus infections do not induce high titer neutralizing antibodies, or at least you don't really see any neutralizing antibodies for about 3 months after somebody's been infected, and then they slowly increase. This is very strange. We say the same thing with the vaccines. You don't get high titer neutralizing antibodies. So we don't know exactly what's protecting these animals, but something is. We don't know whether it's a T-cell response or non-neutralizing binding antibodies, but we know it protects, and we know it's broadly cross-protective. And that's possibly because it's not down to neutralizing antibodies, which tend to be very specific. So the mechanism is different, and that may actually help us with the cross-lineage response, because we would really like to have a vaccine that protects against all the lineages. In further work, Macaques were vaccinated and then challenged, and we see very, very similar results to the guinea pigs. There's a transient viremia, a little tiny bit of virus detected here, but really low levels, whereas in the control group we see a rapid increase in viremia. So this vaccine protects animals, and we need to know if it protects people. So we have manufactured the vaccine in our Oxford vaccine manufacturing facility, where we manufacture the COVID vaccine. And a few weeks ago, we started our first clinical trial in UK volunteers. And we will complete that study, and then we'll be moving to West Africa, to the Lassa fever belt, where we'll be continuing clinical development of this vaccine. What we're seeing so far is very good, because with a single dose, we're getting protection and no viremia. So it looks promising, but we need to test this in people. And because there are so many cases of Lassa fever virus, it's expected that it will be possible to do a vaccine efficacy trial in West Africa, particularly in Nigeria. Now, this does not have a high fatality rate. It has a relatively low fatality rate, but one lasting effect that you could have, if you have a Lassa fever infection as a child, there's quite a high chance you'll be left with deafness. Sometimes this is temporary and sometimes it's permanent. So there are a lot of children in Nigeria who, unless they get a cochlear implant, are left deaf because of Lassa virus. So that's another public health problem. It's not just the deaths and the hospitalizations that we want to prevent, it's the deafness, because that really causes problems as well. So that's one of the viruses that we're working on. Another one that we started early on was Middle East respiratory syndrome. We started working on this with UK funding before CEPI had even been formed. Now, many diseases are held in zoonotic reservoirs, and they can be quite often bats, and they can be rodents, but sometimes they start off in a small animal, and then they move into a livestock species where the amount of virus is amplified, and then because humans are in contact with that livestock species, they are at risk of being infected. So what we think happened with MERS, which is another coronavirus, is that it was originally found in bats in East Africa, and from the bats it spread into some camels. And it started to circulate among the camels, and it adapted to camels. It's not the same virus as was in the bats, but it was circulating among the camels. And it's like having a seasonal coronavirus infection for a small child. The young camels get a snotty nose and they're a bit off colour, and then they recover, and there's no lasting effects. It doesn't kill them, it doesn't really cause any serious problems, but it can spread among the herd. And because there's trade in camels from East Africa into the Middle East, some infected camels moved, and this virus is now endemic in camels in the Middle East. And there are frequent outbreaks amongst the camels, so the young camels will become infected. Coronaviruses don't leave you with lasting solid immunity. You get reinfected, the immunity wanes and you'll get reinfected, and that's what happens to the camels. So the highest viral loads are in the young camels, but then the slightly older camels get infected as well, and the virus spreads around and then it dies away again. And people can get infected if they're in contact with the camels, because the receptor that MERS virus uses to get into a cell, the DPP4 receptor, is very, very similar in a camel and a human. So if it can get into a camel cell, it's the same virus can get into a human cell, and it does this very readily. It's not like Lassa, where it's quite unusual to have human-to-human transmission. Here, goes very easily from camel to human and then human to human. And about 50% of MERS cases are known to have contact with camels. It's a bit worrying that we don't know how the other 50% got infected, because that suggests that it's spreading in the population. Somebody's been infected from a camel and then it's spread, and we've only found the disease later on. But this is because it's really only a dangerous disease in older people, people with compromised immune systems. And if you take blood samples from people who work with camels, you quite often find they have antibodies against the MERS virus, but they don't report being seriously ill. So it's spreading among the population, and if it gets into a hospital, that's when you have big outbreaks. So Saudi Arabia now triage anybody going to their accident emergency. If it's a respiratory disease, you go one way. If it's anything else, you go another, so that you don't have everybody in the waiting room getting infected with MERS virus when you went in with a sprained ankle. And that has helped, and that and other measures have really reduced the number of hospital outbreaks in Saudi Arabia. In South Korea, there was an outbreak where one businessman from South Korea traveled to the Middle East, went to a couple of different countries, went home again unknowingly infected with MERS virus, went to 3 different hospitals, and then a member of staff went from one of those hospitals to another, and all of those 4 hospitals had MERS outbreaks. And this, from one person, this wiped about $10 billion off the South Korean economy. And they dealt with it very rapidly, but one person traveling caused outbreaks in 4 hospitals, and it was not easy to contain. So camels are really an important part of life in Saudi Arabia. They're used for food, they're used for milk and meat, they're used for entertainment, so racing, camel beauty contests, people keep camels as pets. They're a real part of the culture. And for a long time, people couldn't believe that it was the camels that were the source of MERS. And it's true that they haven't always been the source of the MERS virus. This is something that's happened over maybe the last 60 years. So prior to the bat infecting the camel in East Africa and then that camel moving to the Middle East, this was not the case. And people have lived closely with camels for a long time. But now there is this real risk of getting MERS infection from camels. And so this is the camel market, well, the Riyadh camel market, but it's now been moved right outside the city. It used to be in the city and people would come and look at the camels, but now it's been taken a long way outside the city. And there's no tourists, no visitors go there. They were doing lateral flow tests on the camels on the day that I visited, so we knew these camels were not infected. But they were doing lateral flow tests for MERS virus in camels well before we all became familiar with them for SARS-CoV-2 in the pandemic. Very similar concept. So it would be wonderful to vaccinate the camels and eradicate the MERS virus in the camels, but because they don't get ill, there's no incentive. It's really hard to get anybody to be interested in eradicating this virus in the camels. We have done a few vaccination studies in camels, and we can induce an immune response. We did one study where we vaccinated some camels and then we brought in some infected camels from the market and we co-housed them, and there was a slight reduction in the level of infection the vaccinated camels got, but not really very promising. The dose we were using was a human dose, and these are large animals, so there's a lot of optimization would need to be done. Also, we get better results with delivery of the vaccine in other animals, delivery of the vaccine to the respiratory tract rather than intramuscular injection. But do you really fancy giving a camel a vaccine sprayed up the nose? Fortunately, most of the ones we would need to vaccinate are only the baby camels, but they're the size of a cow. So, that hasn't really picked up. But it would be wonderful if we could eradicate the virus in the camels. But again, we are working on a vaccine to use in humans. And this then formed the foundation of the work that we did in the pandemic. So we'd made a Chaddix-1 vector vaccine expressing the spike protein, same concept as the pandemic vaccine. We'd vaccinated people in the UK with 3 different doses, and we saw the peak antibody response correlates with the dose that we'd given them. The high dose is the highest antibody titer. So that's the dose that we used in 2020 for the SARS-CoV-2 vaccine. We see T-cell responses. People have already got some T-cell responses that recognize the MERS virus because we've all had seasonal coronavirus infections, and there is some cross-reactivity, but then it's boosted when we give them the MERS vaccine, and it stays at a high level. A year after vaccination, these T-cell responses are higher than they were before people got vaccinated. So we've now given them a MERS-specific T-cell response. And we get neutralizing antibodies against MERS virus as well, and this is showing you neutralizing antibodies against different strains of the MERS virus, and again, the highest responses with the highest dose. So we'd done this clinical trial in the UK. We'd done exactly the same clinical trial design in Riyadh, in Saudi Arabia. We were talking about taking this vaccine further into development, and then you know what happened in 2020. We all had to refocus. But because we'd been working on this coronavirus vaccine, we knew how to go about making a different coronavirus vaccine. The challenge I had was our manufacturing facility where we made the vaccine, or we can make vaccines for early phase clinical trials. There's one clean room. We can make one vaccine. We've been trying to extend it for years. We've still only got one clean room. So you have one shot. So what we're going to do, well, we're going to do exactly the same thing as we did for the MERS virus, exactly the same design, except it's a SARS-CoV-2 spike protein, not the MERS spike protein, because we got a strong immune response, and that was the best we could do. Changing the design might work better, might not work as well, so we just went with what we knew worked, started on it. As soon as the sequence was released on about the 11th of January, we made the prototype, we got it into our manufacturing facility. Very quickly, we kicked out the project that was in there, fumigated the clean room, started manufacturing this. Our colleagues in Rocky Mountain Labs in the US were doing the vaccination and challenge studies in non-human primates. It was very important before we started our clinical trials to know that if a non-human primate had been vaccinated and then exposed to a large dose of the virus, there would not be a pathogenic response, because there have been a couple of examples in history, particularly with an RSV vaccine in the 1960s, where vaccinated children actually did worse than non-vaccinated children when they then encountered the real virus. Now, we think we understand why that is, and we had absolutely no expectation that was going to happen because of the type of immune response we get with this vaccine, but we had to demonstrate that. We had to show that before we started vaccinating people, you would not have worse disease if somebody who was vaccinated then encountered the virus. And of course, as we all know, that turned out not to be true. But we'd proven it before we started vaccinating people. So we worked with the regulators, the ethics committee. We got approval for our trials, and we started vaccinating in April in a very large study for a phase 1 study. We started with 1,000 volunteers right from the beginning. We expanded around the UK into 10,000 volunteers in our phase 2 and 3 trials. We expanded into clinical trials in South Africa and Brazil. And these were blinded, these were placebo-controlled trials, one-to-one randomization to vaccine, and actually wasn't placebo, saline placebo, it was another vaccine, because we really didn't want people to understand whether they'd had the COVID vaccine or not. And if you give somebody saline, they probably know they haven't had the real vaccine. So we gave them the COVID vaccine or a meningitis vaccine, so they wouldn't know and wouldn't change their behavior. And then we had to wait for cases to accrue. So you have to wait until enough cases have been determined in your study to see if you can then unblind. And then that took months because, as you will understand, countries were going into lockdown, and we were taking measures to prevent people becoming infected. And we weren't telling people, no, just go out and get infected, because we need to know. Everybody had to comply with the regulations. We were originally told by the epidemiologists, the people who were doing the modelling, that if we could get our trial started in April or May, the cases in the UK would peak probably late May, and we'd be well placed to get a rapid answer. But obviously, cases didn't peak in late May. By March, we'd gone into lockdown. So we had to conduct our clinical trials in lockdown, which was a very interesting experience, very challenging to do, but we managed to do it. We didn't have any infections occurring within the trials team or from the volunteers to the trials team, because remember that if somebody thinks they've got COVID, they have to come back to our clinical centre to be tested and have all their symptoms recorded. So we had to manage all of this very, very carefully to make sure they didn't infect anybody else. But we got the case numbers eventually across the different countries. We broke the code. We discovered it was an effective vaccine. We got licensure from the MHRA in December, and vaccine rollout started in January. And this was only possible because AstraZeneca partnered with us in April and put a massive effort into manufacturing in multiple sites around the world. I think we had 25 different manufacturing facilities, and they deliberately made it very low cost and made it around the world so that it could be used around the world. And for that reason, combined with the efficacy of the vaccine and the fact it doesn't need frozen storage, this vaccine saved more lives than any other in its first year of use. There was a lot of it, and it could get to people, so it saved more lives. But post-pandemic, we returned to thinking about these other outbreak pathogens that we still don't have vaccines against. And I want to tell you a few stories about a Rift Valley fever virus vaccine. This is led by my colleague George Warrenway. And this is a virus that infects predominantly sheep, cattle, and goats. But the virus is now endemic across Africa and also in the Middle East. So this is a One Health approach to vaccinology. So most new human infectious diseases originate in animals of some kind. And often livestock are involved in amplifying the disease. They get exposed to some wildlife reservoir, it then amplifies, and the people who work with the livestock get infected from their livestock. When this happens, when the livestock get infected, there's an economic impact of death and disease in the livestock, potentially trade embargoes because people don't want to be trading infectious livestock. There's a reduction in food security. So all of these are things that threaten human health. The economic impact is ultimately bad for human health. People can't afford to eat or to afford medical care. Reduction in food security is clearly bad for human health. And we also get spillover. So spillover is what we call it when viruses move from the animal reservoirs into people. And this happens all the time, and often there might only be 1 or 2 cases and the virus doesn't really take off in humans, but with Rift Valley fever virus it does. And so we need to try to stop this happening. But understanding the disease in animals, in the livestock, helps us protect humans both directly through vaccinating them, but also indirectly through vaccinating the livestock. Now, this is a disease that causes serious effects in the livestock. To complicate matters further, it's carried by insects, and not one very specific species. It can be carried by a number of different insects, and the infected insect eggs can stay in the ground for up to 10 years if it's dry. And then when the rains come, the eggs hatch, and they're already infected, so you then have adults carrying Rift Valley fever virus, and the grass grows because it's rained, and then the livestock come to graze, and they get bitten by the infected insects, and they get a Rift Valley fever virus infection. And it's very hard to predict where it's going to happen because of this fact that the insect eggs can lie dormant for long periods of time. But these can infect livestock. In sheep, the most obvious sign of this is spontaneous abortion, because obviously flocks of sheep are all pregnant at the same time. This disease is not particularly dangerous to adults. It has high rates of neonatal mortality if the lambs get infected, but if the pregnant sheep get infected, they lose their lambs. So a farmer can have almost no lambs from his flock of sheep in a given year because there's been a Rift Valley fever virus outbreak. In humans, it causes a range of symptoms, including abortion. It's also picked up in women at maternity hospitals who have fever, and it can be fatal. It also infects wildlife, but humans can get infected directly from the infected insects or from their contact with wildlife. So the farmers who are working with the wildlife are at risk of this disease. Now, there is a licensed vaccine that's used in sheep in South Africa. It's called Rifax, and this is a live attenuated Rift Valley fever virus. So it's derived from the pathogen itself in the way that some human vaccines are. It's not entirely safe in the animals. If you vaccinate pregnant animals, some of them will lose their lambs. So the farmers have to choose between vaccinating their flock, possibly losing, well, they will lose some lambs, or not vaccinating. And then if there's a rift outbreak that year, they could lose all of them. So it's a gamble. So what we want is a vaccine that protects the animals without causing any losses in the pregnant animals. And so this study was set up, first of all, to test it in young animals. So Chanox-1, Rift Valley fever virus, the Rifax, the licensed vaccine, and then saline control, with sheep, cattle, and goats, young animals who were vaccinated, and they developed antibodies, and then they were exposed to Rift Valley fever virus, and their viremia was measured. And what happened was that all of the Chanox-1 vectored vaccine animal groups developed high titer antibodies against the virus. Most of them with the Rifax, the licensed vaccine, did. 1 sheep and 1 cow didn't make antibodies. And when they were exposed to the virus, those 2 who hadn't made antibodies became viremic, although they survived, and all of the animals who got saline became viremic. They also survived. These are not very young animals, so they can deal with the infection, but this is the first test of the virus in these animals. So this shows you the antibody titers here. Blue is the Chanox vaccine. Black is the Rifax vaccine. You can see there's 1 cow, 1 goat that didn't seroconvert. And when they're exposed to the virus, all of these animals with antibody titers that are measured were protected, but these ones weren't. So a good start. It was also tested in pregnant animals, and the vaccine itself, the Chanox-1 vector vaccine, did not cause any losses in the pregnant animals. There were some losses when they were then exposed to Rift Valley fever virus, but very few, whereas the Rifax vaccine itself caused some losses. This has now gone on to do a a larger trial in these animals, including camels. Camels can also become infected, and because they're used for transport and they travel long distances, they can actually spread outbreaks. So we need to keep an eye on the camels and making sure they're not taking the virus from one place to another. So these animals were vaccinated to look at their ability to develop antibody responses. These weren't then challenged with Rift Valley fever virus, but their antibody responses were compared to the licensed vaccine to see if they were as good as the licensed vaccine. And this trial is still ongoing. And these are some of the animals that are being enrolled into the study in the environment in which they live. Quite remote places, but the farmers do vaccinate their animals, and so if we have this vaccine developed, then it will be possible to protect them against Rift Valley fever virus, and it protects the farmers as well, both from direct infection and loss of their livestock and the economic impact that comes from that. So clinical trials in people as well. As you might expect, we see good antibodies. It correlates with the dose they've been given, and this is now going on into a larger study to look at larger numbers of people in different countries, UK, Uganda, and now in Kenya. So for the Rift Valley fever virus vaccine, we see high levels of efficacy, protection against disease and fetal loss in the pregnant animals, and safe and immunogenic in humans, now going on into test it in larger numbers. And I need to hurry up now to tell you about the last virus for this evening, which is Nipah virus. This is a very scary virus because it has a very high fatality rate, average 72% fatality of everybody who's ever been infected with this virus. First identified in Malaysia, where it infected pigs. And what happened, this is another virus in bats, fruit bats this time. They were dropping contaminated fruit near the pig pens. The pigs were eating the fruit. Pigs got infected, spread amongst the pigs. Then it spread to humans. Big outbreak of a virus never seen before, spread to a number of different countries, was contained in Malaysia by culling all the pigs. It was the only way to end the outbreak. Subsequent to that, most of the cases have been in Bangladesh, and this is because the fruit bats like to get into these pots that collect date palm sap, because people like to drink the fresh date palm sap, unpasteurized, and this is one of these pots, and you can see the bats, because they like it as well, and they get into the pots and they contaminate it, and people get Nipah virus infection from drinking the infected date palm sap. Now, there has been some work done in simple interventions, like cover the top of the pot with this bamboo skirt. This works quite well, but we still see Nipah infections all the time, and it's a paramyxovirus of a type. We don't have any vaccines against it, but we do have a vaccine against a very closely related virus identified in horses in Australia, again, moved from the fruit bats to the horses, Hendra virus. People got infected through contact with the horses, 7 human cases, 4 deaths. So again, quite a high fatality rate. A vaccine was registered and used in the horses. These are large and expensive animals, and it's a fatal disease in the horses, so people wanted to protect their horses, and by protecting the horses, people don't get exposed, people don't get infected, and it stops Hendra virus spreading. We don't have that situation with Nipah virus because most of the cases don't involve livestock. It did in Malaysia, it was pigs, but in Bangladesh and in India, where we see cases, it's generally contaminated date palm sap or fruit that people consume and they get infected. So we've been conducting a clinical trial. We've tested 1 dose of the vaccine. This is a good antibody response after 1 dose. 2 doses, it goes even higher. We think this is probably enough to protect people in an outbreak, but this might be good for the surveillance teams or the healthcare workers where we want really long-term protection, and the Ceylon group didn't develop any antibodies. So we now have a development plan to take this through to licensure. So we've already done the proof of concept preclinical studies. We've manufactured the vaccine, we've started our clinical trials, we've worked with the Serum Institute of India, who are now manufacturing this vaccine at large scale, as they did. They were one of the manufacturers for the COVID pandemic. So we're getting ready to go through process validation and all of the manufacturing parts that we need to do to get this vaccine licensed. And we will do a larger study. We've done the UK trial, we've started a small trial in Bangladesh. We will extend that trial into larger numbers of people. But we need to know if it works. And we're not going to know that from humans, most likely, because the outbreaks tend to be small. They're spotted easily because there's such a high fatality rate. So this is where we need to use animals to do what we call immunobridging. And the way this works is that, first of all, we vaccinate non-human primates with a range of different doses of our vaccine, going down to quite low doses so that we get a range of different antibody titers. And then we challenge these with the Nipah virus from Bangladesh, and we see which of these animals are protected. Now, we expect that the ones at the highest dose will be, but somewhere as we go down the dose levels, we'll start to lose that protection. That enables us to have an estimate of what antibody titer they need to protect the animals. And then what we do is we vaccinate some people, we plasmapherese them, and we purify their neutralizing antibodies, and we transfer those into non-human primates at different dose levels. And we'll do a first study where we give them 4 different dose levels, 5 different dose levels in a control group. And then we challenge these animals and we determine the cutoff, what is the level of human antibody induced by vaccination that will protect these animals. And then that will lead to statistical design of another study. So this is designed to get really solid, statistically valid information with the smallest number of animals. That's why we do this in 3 steps. And by the end of this, we will be able to say that this level of antibody protects against Nipah virus infection, and we can compare that level of antibody to the level that we're inducing in the people that we've vaccinated, and provided the level in the people is higher than the titer known to be protective, we can get emergency use licensure for this vaccine, which can be stockpiled, and if there is an outbreak of Nipah virus in the future, we will have this ready to protect people. Now, Nipah virus is a close relative of measles virus, and measles virus is known for being the most transmissible virus there is. Currently, Nipah virus is not very good. It does transmit between people, it doesn't do it very efficiently. But if we allow it to keep practicing, viruses mutate and they get better at transmission. We saw that in the pandemic. The more time the virus gets the opportunity to move from one person to another, the more likely it is there'll be a mutation that increases that transmission. And if that happens with Nipah virus with a 72% fatality rate, we are in big trouble. So we need to stop these outbreaks. We need to contain them really, really quickly. And having a vaccine stockpile, such if there was a larger outbreak, having a vaccine stockpile that we know is effective against the virus would put us in a very strong position to contain the outbreak, protect the people in the outbreak area, but also protect the rest of the world, because we really do not want a Nipah virus pandemic. So we need to get the vaccine ready now. So that's where I'll stop. I'm not going to acknowledge individual people because there have been so many of them. I do want to acknowledge all of the volunteers that take part in our clinical trials, and there are many of them. Our funders, of course. And this is done at the University of Oxford with many partners around the world, and we continue to try to build and strengthen those partnerships because we all need to work together to get the best protection we can against these outbreak pathogens. I'll stop there. Thank you.
Phenomenal lecture. Thank you so much for all that you do to keep the worldwide public healthy from current outbreaks and future ones. Thank you very much. I think we can open up the floor for any questions for our lecturer tonight.
Thanks. Hello. Oh, now it is. Sorry. It's a really fascinating tour through virus and vaccine development. Just a really naive question. Why is it that non-human primates are used for the vaccine trials?
Because they're the most similar to humans that we can get. And some of these viruses don't infect other animal species. Some of them infect lots of different animals. Like Rift Valley fever virus infects lots of different animals. Some of them are really very specific to primates. And so we need to use non-human primates. We don't use the non-human primates until we've done everything that we can, sometimes you can use a transgenic mouse to get some early, but that's a very artificial situation. So if we're going to do this licensure by immunobridging, which we don't think we need to do for Lassa, because there's enough cases we can do an efficacy trial, but for Nipah and for MERS, it will be licensure by immunobridging. The regulators want to see performance in a close relative of ours.
And can you imagine what a non-animal method might look like, a replacement technology that could replace non-human primates for these studies?
So there's quite a lot of work going on with human organoids, particularly using tonsil tissue. So if somebody's having their tonsils removed for, because they need their airways clearing, you can take the tonsils and disassociate the tissue and then put it into culture, and that starts to form something a bit like a lymphoid organ, and you can vaccinate it and see what the immune response is to that. However, it's only good at looking at immune response, amplifying an immune response that that person already had. So we've all been exposed to flu. So if you give a tonsil organoid a flu vaccine, you can amplify their pre-existing flu response. But if you give them a Nipah vaccine, it does nothing because they don't have a they don't have a memory response to Nipah virus. So currently, we can look at boosting responses that the person whose tonsils removed had already got. We haven't got anything yet that allows us to look at priming new responses. I think it probably will come. People are trying to make artificial ways of setting up human lymphoid systems using blood cells, but we're not quite there yet.
Thank you.
Thank you, that was fabulous. Probably nothing to do with the research that you're doing, but bird flu at the moment, every day we're having sort of emails coming out from DEFRA saying there's another breakdown in a poultry farm and obviously destroying loads of birds. So is there any way that something can be produced for that?
So we are actually doing some work on H5 flu in cattle with the Royal Veterinary College. We've vaccinated some dairy cows, and we're looking to see, do they make antibodies in the milk as well as in the blood? So that study's ongoing at the moment. Flu is so difficult because there are so many different variants of the flu virus, and it's a real challenge. So with the H5 and the poultry farms, then it's very difficult to vaccinate all the poultry because you need to do it really quickly, and actually chickens at poultry farms already got a lot of vaccines, but they have to be really inexpensive to make it cost effective. I'm concerned about the infections that have happened in some countries in cattle. This is novel. We haven't seen H5N1 infections in cattle before, and that's then spreading to cats on the farms, and cats go into people's houses. So, I mean, I think that's something that really needs a close eye keeping on it, and I'm not sure that's happening. We probably will see some bigger outbreaks of H5N1 in the future because it seems to be spreading into more and more species. It's not just in the poultry now, it's moving into mammals. And it's a bird virus, it's an avian virus, but the fact it's adapted to cows and then it can spread to cats shows us that the virus is changing and adapting and getting better at living in mammals. And that's what we shouldn't give viruses the chance to do.
Thank you very much for that. It's fascinating. Do you think that we are better prepared for a future pandemic than we were pre-2020?
On the whole, no. I think a few people, I mean, we learnt a lot from what happened, but that learning doesn't spread very fast, certainly doesn't spread to politicians. Politicians are in a difficult situation because they have a very short tenure in office for the most part, and so they're not interested in you know, what's going to be best for us in 10 years' time. And we've seen the reports on the COVID inquiry and how that wasn't really managed very well. We're not really better prepared. There's still an awful lot that we could do, but we should be doing the research. It's a lot cheaper to do the research, to have the vaccine stockpiles, to have the diagnostics ready to go, than it is to have to deal with a pandemic when it happens. Of course, everybody's hoping it's not going to happen anytime soon, but it could happen next week. We don't know. It might not happen for another 20 years. But the better prepared we are, then the quicker we can deal with it. And the fact that these viruses exist in wild animals, in the rodents, in the bats, in wild mammalian species, we are seeing more spillover events because of deforestation, of humans taking over more of the world. There's more contact with the animals, and that means more contact with these diseases, and so there's more opportunity for them to spread, and we need to be ready to respond. And I don't think we're very much further on than we were from 2014.
Thank you. Thank you, that was great. Are there any of those, or maybe some others that you haven't talked about, that really keep you up at night? Are there any in particular that worry you?
Well, flu. I mean, because, flu spreads so rapidly. You go around the world in days. If you get a new strain of flu virus that we haven't been exposed to before, it goes so quickly. And it will infect all of us. And in 1918, the fatality rate was under 2%, but it killed 40 million people because it just infected everybody. And that's so 2% fatality rate. If it infects a lot of people, that's absolute crisis for healthcare systems. That completely brings everything to a halt. It may not sound very worrying, but if it's a virus that spreads rapidly and everybody's infected with a virus that has a 1 or 2% fatality rate, that just brings an end to the ability of hospitals to deal with it. And so, yeah, flu's the one that's, I think, is the most worrying.
Really interesting talk, thank you. I was just curious on how there's different viral pathways, there's different vaccines you can kind of create. Has there ever been any challenges with what models you then test the vaccines on and whether they're compatible?
Sorry, with what was that last part?
With whether they're compatible, like have you ever found that there's been a vaccine that's not compatible in an animal you've tested it in, but maybe work with humans?
So we did some work with bats, and we got some immune response in bats, but it was hard to know how good it is because people aren't very practiced at studying immune responses in bats. So I don't know that we'll ever be able to vaccinate the bats, possibly, but that would need a lot more work. Other than that, we do see really good immune responses in all the animals. We've used it in chickens. So yeah, we could vaccinate chickens against H5. It's just a question of whether it's economic to do that. Elephants, we've vaccinated, well, not us directly, but we provided vaccines to a group that wanted to vaccinate elephants, and that seemed to work quite well. So yeah, generally, the adenoviral vector vaccines are very good in a wide range of species.
I've got a question about communication and understanding. Obviously, the pandemic was dreadful, but the one thing that we all told ourselves afterwards was at least now everyone will understand the value in animal research for good, and people will now understand the value of vaccines for good, and yet 4 or 5 years later we find ourselves in a very different position. You've won a lot of plaudits for the way in which you communicate your science and the way that you talk about what you do, and yet we're still in this position. So what can scientists do about that?
Well, that's obviously really very, very difficult. There's a number of things that we try to do. So one is just providing accurate information. So there's a website called the Vaccine Knowledge Project, which was originally set up to provide information about paediatric vaccinations so that parents could go and get trusted information for themselves. Because actually some healthcare providers are not very good. Some midwives are quite anti-vaccine, oddly. But this was a way of providing information, accurate information, written in lay language to people who could read about all the vaccinations that have been recommended for their child and understand, the value of them and when they should be given and all of those things. And we added to that during the pandemic, and we tried to make it more global. So making accurate, trusted information available, knowing that there's somewhere where you can find the facts as opposed to what somebody on social media said about it. That's one thing. I think something we need to do more of with schools is actually training school children to assess information that comes their way. So just because somebody on social media says something, that doesn't mean it's true. So what do you do about that? How do you start to think about, well, who's giving you this information? What's their agenda? What's the reference? Can they substantiate it? Where's this coming from? Actually, just because there's been such an explosion of social media, certainly during my lifetime, But a lot of the information that's out there is political, and it's from somebody who has their own agenda, and you can't trust it. So how do you separate the facts from all of this things that people are telling you when they really don't know what they're talking about? Why should you trust a pop star telling you which vaccine to take? You know, should you not think about going to somebody with some medical qualifications instead? So I think that's something that we need to do, particularly for young people, is to start to give them the ability to look at where they get their information from and what to make of it.
That was really great, thank you. I was just wondering about we're seeing a lot of cuts to sort of international aid programs from the likes of the US and the UK. Do you see that having an impact on vaccines and the future development of vaccines?
It's certainly having an impact on the deployment of vaccines. So Gavi, the Global Alliance of Vaccines and Immunizations, have a lot of their funding cut. And so I was at a symposium in Brazil when these cuts were being announced, and people there were immediately saying this is going to cause 1 million child deaths because of existing licensed vaccines, that now the money isn't there to get these vaccines to the children that need them. This isn't new vaccines, it's the existing ones. And there are more and more children in the world who have not had a single vaccination. With all the vaccines that have been around for decades in some cases that we know are safe and effective and will protect them, and children just aren't getting vaccinated. So that's one problem. We haven't yet really seen a cut in funding for the development of these outbreak pathogen vaccines, but then there wasn't ever very much funding there in the first place. So it will be hard to see much of a cut. It is still happening, but these vaccine development programs get more expensive as we get later stage. You know, we start off with small clinical trials, then we go into larger clinical trials, and these start to get a lot more expensive. So to get all of these outbreak pathogen vaccines through to licensure does require not only scientific collaboration, but funding collaboration. And CEPI, the organisation that fund a lot of this, are now saying that they don't have pockets that are deep enough to fund all these efficacy trials that we need to do. So they're seeking co-funding from other organisations, that when you have a vaccine that looks very, very promising in a particular region of the world needs to have this vaccine or a stockpile of this vaccine ready to go, that they will put in some of the funding that's needed to finish the development.
Hi, thank you for the lecture. I would like to go back to the One Health concept and how vaccine science as to kind of go into a dialogue with other areas of science, be it transdisciplinary, especially with climate change and also with any ecological issues, as you were saying. How do you think you envision this kind of collaboration going between like the more hardcore vaccination science and vaccine science with these other types of influences that may condition flows of infection, for instance?
Yeah, well, that's really difficult, and personally, I haven't had very much success with trying to engage people about vaccinating their camels, for example, because their camels aren't ill, so why would we vaccinate them? And people don't want to. There's another disease called Crimean Congo hemorrhagic fever, which is found, this is more Eastern European and around the Mediterranean, and this infects sheep and goats as well, but can also be transmitted to humans, where it causes a hemorrhagic disease. It's tick-borne. It's carried in these horrible ticks that are called hunting ticks. They will chase after you. We think of ticks as kind of sitting on a leaf, and if you brush against it, you might pick up a tick. But these are hunting ticks. They actively seek out warm-blooded mammals and attach, and they carry this disease. And so they infect the livestock, and they also infect humans, and humans get direct infection from the livestock. And again, this is another virus that doesn't really cause any symptoms in the animals. So nobody's interested in trying to vaccinate the animals. And why would you have a big vaccination campaign to vaccinate the animals when they're not getting sick? But that will be the way to contain it, because if we don't do that, it's going to keep infecting the animals, and we're going to keep getting spread into humans, and then you can have an outbreak in humans. So in the cases where you've got a valuable animal and a fatal disease like Hendra in horses, very easy to make the argument. The horses are going to die because they're grazing on infected pasture that the fruit bats have contaminated, and they get sick, and then people get sick. So we can vaccinate the horses, and we protect the humans. Everybody wants to protect the horses. These were race horses, very valuable. and a vaccine is available. But that's not true of sheep who aren't even getting sick. So the challenge is to persuade everybody that when we've identified a situation where diseases are regularly spreading from livestock, we need to suppress it in the livestock, and then we won't see the human infections.
Okay. Dame Gilbert, thank you very much for a fascinating talk. And well, can we please thank our speakers for tonight? Thank you.